Hello and welcome everyone. My name is Stéphane Horel and I'm an investigative journalist at Le Monde, specializing in corporate influence on science and corporate harm. I will be the moderator of this webinar today, which will focus on the COVID-19 vaccine race. We're now living in Groundhog Days of bad news. The world waits for an effective and safe vaccine to mitigate the far-reaching impact of the COVID-19 pandemic, as a number of vaccines enter the last stages of their accelerated trials. Expectations are very high, but developing a safe and effective vaccine, especially in such a short time, is fraught with difficulty. Science is not neutral. Science is not like the Shire in The Lord of the Rings, and scientists are not hobbits with a pure heart, looking for the truth wrapped in the white coats of objectivity. Like any social group, scientists are caught in the middle of social tensions, career considerations, and also commercial interests. How can journalists report these developments in an accurate and knowledgeable way, especially as the pandemic has forced many of them to become healthcare experts overnight? Not easy task. This is what we will be talking about today, and we have two excellent speakers. Now there's a special reason for this webinar on the COVID-19 vaccine race. It's the second event to mark the publication of GIJN's new guide, Investigating Health and Medicine. This 90-page manual covers such issues as the COVID-19 pandemic, drug development, medical studies, assessing evidence, corporate influence, and conflicts of interest. In this webinar, we are pleased to bring together one of the co-authors of the guide and a scientist whose research includes a focus on the drug approval process. Together, they will show what red flags to look for, both before and after a vaccine is approved. The speakers will also explain how to assess a scientific study and will share tips to avoid common pitfalls in medical investigative reporting. So meet Serena Tinari. She's a medical investigative journalist and a co-founder of GIJN, a Swiss non-profit organization dedicated to investigating and mapping health affairs. She specializes in drug safety, evidence-based medicine, and conflicts of interest. A trainer, speaker, and moderator, she has reported extensively on swine flu and Tamiflu. As noted, she's one of the co-authors of the new GIJN guide. Hi, Serena. Peter Doshi, PhD, is an associate professor of pharmaceutical health services research at the University of Meryland School of Pharmacy and associate editor at the British Medical Journal, the BMJ. His research focuses on the drug approval process, how the risks and benefits of medical products are assessed and communicated, and improving the credibility and accuracy of evidence synthesis and biomedical publications. Hi, Peter, and thank you for giving some of your precious time to us journalists. Just before we start, a little bit of information about the Global Investigative Journalism Network, for those of you who are not familiar with it. GIJN is the largest global network of nonprofit investigative journalism organizations, with 203 member organizations in 80 countries. But it works with journalists everywhere, in nonprofits, in commercial organizations, and with freelancers. It has an extensive range of resources and tip sheets to help journalists worldwide, including, as I said, the new guide on investigative health and medicine, investigating health and medicine. So please look out for that and also check out the many resources at gijn.org. We also want to hear from you in the audience, so please send written questions and messages in the Q&A box, and we'll answer to as many as we can. You can start asking questions from now, and when the speakers are finished, my GIJN colleague Eunice Au will join us on screen to moderate the questions. So let's start now. Over to you, Serena.
Yeah, good day, everyone. And thank you for having me. I will now share my presentation. So as Stéphane already mentioned, this is GIJN, is the non-profit organization I co-founded together with Catherine Riva. We are both medical investigative journalists, and we work with methods, tools, and ethics that are really at the crossroads between investigative journalism and evidence-based medicine. And they share really a similar approach and also kind of a thinking. That's why we think it's really good for investigative journalists and journalists, generally speaking, to stick to evidence-based medicine principles. So this has been, he is still a very difficult year for everyone, but I need also to say that for a medical investigative journalist, it's an especially bad year. It's actually a nightmare because as Stéphane has mentioned already, and we all know it, literally overnight, virtually every journalist on the planet had to improvise becoming a healthcare reporter. And actually the problem is that investigating healthcare is really complex and challenging, and the learning curve can be steep. So becoming suddenly a medical journalist, as many had to do during this COVID-19 crisis, it's full of pitfalls. So I will admit here that I feel a lot for all my colleagues that find themselves in this difficult situation, but we can definitely do better. I have to say that pandemic journalism until now didn't do a great job. It's possible to improve our journalism in this field, and it's also something that is really in the public interest, because what we are doing today has really a lot of impact on policies, on governments, on people. So we need really to work on this. And that's why we have the guide. We have the GIJN guide. I will introduce you quickly to where you find in the guide the knowledge to also read the COVID-19 vaccines race. This is the guide cover. The guide has been published and commissioned also by the Global Investigative Journalism Network. It's open access, so you find it on GIJN website. This is the table of contents. The idea behind the guide is really to provide knowledge and tools to independently assess the evidence, critically appraise the risk-benefit ratio of any given product or policy, and expose corruption and malpractice. So there are many, many tips. It's 90 pages with like 500 resources. So you can use it as a manual, reading it as a book, or you can just pick the chapter that can help you in your work as a journalist. So we also have, of course, a chapter, a preface actually, about the COVID-19 crisis. And it's like more like tips and principles that we encourage journalists to keep in mind when reporting about COVID-19. And among several tips and tools we wrote when reporting about COVID-19 vaccines and drugs, be extra cautious. And this is the topic of today's webinar, of course. So quickly, we give several tips in that chapter, but these are probably the most important. So beware of oversimplification. This is a very complex field and you can't simplify it that much. Be very cautious about the models, because this crisis has been really driven by amazing modeling of what will happen. And never forget that models are like predictions, so they should always come with a warning. Stick to the best available scientific evidence. You could tell me, well, of course, it's not really what is going on right now all around the world. And of course, to stick to the best available scientific evidence, you need first to understand what is that. And in the guide, you find quite a lot of knowledge to help you there. Provide context, because no number can make sense without a denominator. So without the context, and in this crisis, really too often, journalists are not providing context. So you need to know what is normal to say that something is really abnormal, for instance. Don't fall for the hype. That should be also logical, right? But it's happening, I'm sorry. And beware of media reporting, because by copy-pasting other colleagues' work, we are not doing really a service to the public interest. So there is more, and in the guide you can find much more. So about the vaccines race, how the media is covering this. These are some headlines, specifically about the Pfizer vaccine. And they share a very important thing, that they were all based, these are Moderna vaccine, the AstraZeneca or Oxford. I mean, they're all based on press releases. So the source of this journalism was actually a press release. And in the last days, since we got this news of UK starting very soon to distribute the Pfizer vaccine, actually, the source of the journalism is government press releases. So I think we all agree that a press release is no evidence, and we shouldn't use press releases as a source for our journalism. So all these articles with, of course, here and there differences, basically they all report efficacy and safety based on the pharmaceutical companies' press releases. And some of them do mention the lack of data, or like there is no access to any data right now. But actually, most of them, they don't really make a big fuss about this. And this is wrong, because we should all be actually concerned that we do not have access to the data. And typically, most of these articles, they do frame the issues as like, when will we get this vaccine? Who's going to get it first? These are the line of questions that they try to address. And these are not really the key and very important issues, actually. So let's have a bit together quickly, a look at some of the issues that we should really look at as a journalist. So in the guide, you find the knowledge you need to read the current situation, especially in the preface, in the chapter 1, and in the chapter 2. And in chapter 2 is the chapter about a study is not just a study. And this is super important because there are major differences between different types of studies. And you find really everything in the guide. And I encourage you to keep in mind what an amazing book said as a line of really reasoning about this. Statistics can pull out of the bag almost anything they may be wanted. That's a really nice book. We encourage you to read it, How to Lie with Statistics. And actually on page 13 of the guide, you find this scary slide. Peter Doshi will show you a similar one later on, because I've seen already his presentation, and he's going to go into more details. But in a nutshell, this is how we normally get drugs and vaccines approved. So they are developed following these stages that are like fixed and is a standard. And they come to the market, as you can see, at the end of phase 3. Is where we are now, actually, with these vaccines. We could talk for hours about this slide. For you, important to see is that anyway, of course, the knowledge about the safety and efficacy of a drug, it's coming with the time. We need a lot of time normally. As you can see, we need almost 10 years at least. So what is going on today, it's something kind of different than this. So this, you need anyway to be knowledgeable about this, to understand what is going on now. What is going on now, as you all know, is an accelerated process. And it has many names, but anyway, it's the big family of the fast track. And originally, fast track was invented with a noble purpose at the end of the day. So to quickly bring new treatments to patients that would address unmet needs. So it's a special procedure that was introduced that accelerates approval by reducing the amount, the quality of the evidence that the industry must submit. However, it's good to keep in mind that in the recent years, fast track became a shortcut for pharmaceutical companies allowing them to skip or shorten in-depth assessment by the regulators. So you need to keep this also in mind. In chapter 1, you find really all the basics about development and approval. So how do we regulate actually drugs? How does it function? And there you can get knowledgeable about some very important points, which are the study design. So how is this study done? And most importantly, the trial protocols. And this will be a lot the topic today, because any trial, any study needs, first of all, to set the aim. What are we going to measure? What do we want to measure? So what is the end point or outcome we want to measure? And this is super important. So about these vaccines, that's the most important information we need. And the good news is that most of the study protocols of these COVID-19 vaccines are public. You find a really good collection on the Riot website. We will put this link also in the chat. And this is extremely important because in the study protocol, you find all the trial protocols, you find all the information. And what is very strange is that there was a lot of coverage about this vaccines race, but journalists didn't go and check the study protocols. So one question, you shouldn't give anything for granted in this situation. I encourage you to read this article by the Center for Evidence-Based Medicine. They have this website called COVID-19 Evidence Service. This is a good article pointing at one big problem we have, that there is a lot of difference how different countries define what is a case of COVID-19. As you know, COVID-19 is the name of the illness and SARS-CoV-2 is the name of the virus, even though everyone calls this the COVID-19 pandemic. This is not exactly accurate. So even the definition of COVID-19 case has many different colors. And then you need to spot the red flags and you need to ask questions like, how is infection defined? So when you go and look at the protocols, because you need to see what is behind the press release and what is behind the hype actually, how is the infection defined? And this is also very important. What are the characteristics of those included in the trials? The age, the pre-existing conditions, because we know that this virus can be dangerous for elderly with 2-3 comorbidities. So this will be, for instance, a key thing. How many of these trials participants were actually that kind of patient or population? So you find all this information in the study protocols, but most importantly in the data. And the big problem we have today, we still don't have data about this study. But this is again the most important question. So what is the outcome that these trials were designed to measure and assess? So was it severe COVID-19? Was it like preventing the infection? And this, you find these answers in the trials protocols. Because I mean, basically, ideally, what do you want an antiviral vaccine to do, actually? You want to do, first, reduce the likelihood that you will get severely ill and go to the hospital and maybe even die. And 2, we want such a vaccine to prevent infection. So interrupt the disease transmission. So are these trials actually designed to answer these questions, to do this? And we have a big problem here. And now is a spoiler alert because Peter Doshi will talk now about this, but I can tell you already that unfortunately, no, these trials are not designed to give us these answers. And we have a big problem, of course. And as a journalist, for me, it's also a big problem that all this media coverage is not addressing the only important question. So that was my side of the story. Over to you, Stéphane.
Thank you so much, Serena, for a crystal clear presentation. And now let's cross the Atlantic to hear Peter Doshi. We're jumping from the European Union to the United States regulatory ecosystem. The drug and vaccine marketing rules of the Food and Drug Administration, the FDA, in the US, and the rules of the European Medicines Agency, EMA, in the EU, have more similarities than differences. More importantly, what they decide has a global influence. So if you're reporting from Peru or Congo, the FDA and the EMA's decision will most likely affect your countries. Peter, over to you.
Great, so thanks for the invitation. Good morning, good afternoon, good evening, wherever you are. I am going to talk, my talk is about the COVID-19 vaccine trials. And so let me now try and get my slides going here. Share my screen, and hopefully you should be able to see them now. Okay, well, interrupt me please if you can't see them for any reason.
We can see them. Thank you, Peter.
Great. So I really want to start my talk off with addressing why these phase 3 trials are so important. And a key reason is that phase 3 trials test a hypothesis. And a hypothesis is a specific research question about efficacy. So said another way, these phase 3 trials, they try to give us our best estimate of how well a product works. They'll also give us some information about the product's safety, the adverse events, side effects, but generally not any long-term data on safety unless the studies are allowed to run long-term. In terms of the COVID vaccines, the trials are testing a hypothesis that the vaccine reduces the risk of getting symptomatic COVID by at least 50%. And if the results show that they do, then people will conclude that the trials proved what they hoped to demonstrate. I'm going to go back to that, but I just want to put that out there right from the outset. And I also want to set the stage by having you, the viewers, think about whether phase 3 trials will tell us whether a vaccine prevents hospitalizations or deaths. So I'll come back to that again. But for now, just, you know, on your own, write down a brief thought about your answer to that question. So how did we get to this point of these phase 3 trials? What you're looking at in this slide is an overview of the pre-approval process for vaccines. Before regulators approve a vaccine or drug, it goes through a series of tests. And some of these happen in test tubes and in animal studies. The key human trials, however, are divided up into 3 steps that are called phases, phases 1, 2, and 3. And that's where we are right now with the vaccines under development. Some are in phase 1. Those are ones we don't hear about as much. Some are in phase 2, which then you start to hear in the news. And the ones that are most widely talked about are vaccines in phase 3. And perhaps the most important thing about this period of time is that it may very well be the last chance that we have to see how the product works compared to placebo. And the reason for that is once a product is approved or authorized, the argument will be made, or is often made, that it would be unethical to randomize people to a placebo since we have an effective product. And so phase 1 are these first in human trials, and they really aren't out to consider much about how well the product works, those phase 1 trials. Phase 2 and 3 trials are where you start to see these discussions about the product's efficacy or how well it works. And I have this slide here because the distinction between phase 2 and phase 3, I think, is quite important to understand. And an easy way to capture the distinction is to think of phase 2 as an investigation into promise and potential, whereas phase 3 is where one seeks proof of your hopes, proofs of that promise or potential. So a phase 2 study, therefore, might be set up to see whether a vaccine can elicit an antibody response. But ultimately, the antibody response is not what we really want from the vaccine. We want something that benefits how people feel, function, or survive, some kind of tangible benefit that I'm calling clinical efficacy. And so it's that phase 3 study where you can actually test to see whether that tangible benefit of clinical efficacy exists and how large or small that benefit is. To quote the FDA, phase 3 studies are large-scale trials to provide a more thorough assessment of safety as well as a definite assessment of efficacy. And to further reinforce this importance of the distinction between phase 2, where we get a potential, a promise, and phase 3, where we have an opportunity to get a definitive assessment of efficacy, and really understand now why that phase 3 is so crucial, I want to share 3 quotes from this FDA document. Now, the context for this document is it was published shortly before President Trump took office, at a time when there seemed the very real possibility that a Trump administration might appoint FDA commissioner that was opposed to phase 3 studies. And that person with holding the belief that phase 3 studies only slowed down the public's right to access medicines. And so this document was published to try and counter that view and really explain why phase 3 trials are important. So here are 3 case studies from drugs that never made it to the market. The first drug is an anti-tumor drug, which in phase 2 showed promising anti-tumor activity. But when it got to phase 3, the drug failed to improve overall survival of patients. Not approved. And there's 2 vaccine case studies in this document as well. One was a herpes simplex virus vaccine. In phase 2, it had positive biomarker results. However, in phase 3, they found that the vaccine did not prevent genital herpes. And here was a staph vaccine, which produced an antibody response in phase 2, and that antibody response was sustained for 1 year. So everything seemed very promising. But when it came to the phase 3 study, the vaccine did not prevent staph infection any better than placebo. And so the lesson here is that things can look promising, and one can reasonably think that therefore a product should work. But when it's put to the test, it might ultimately fail to deliver. So the lesson is you can't guess clinical efficacy, you need to demonstrate it. So for COVID vaccines, what specific question about efficacy is being tested in the phase 3 trials? What are those trials aimed at answering? Just what will it mean when there's an announcement, as we've seen, that we have an effective vaccine? What does it mean? To answer this question, I want you to consider the nature of COVID disease, and then have you think about what you would expect of a vaccine. And what this COVID-19 iceberg captures is that disease occurs along a spectrum. Everything from infections that cause no noticeable symptoms at the bottom to mild symptoms that don't require seeing a doctor, all the way up to the most severe outcome of death. And note that I'm calling this an iceberg, not a pyramid, because the bottom numbers are many, many times larger than the top. But even though those numbers at the top are much smaller than those at the bottom, the deaths and hospitalizations have higher public health relevance. We're not concerned about COVID-19 because many people have mild symptoms. We're concerned because of the hospitalizations and deaths. So now the question is, what do you want your vaccine to do? And I imagine most people, certainly myself, will say that you want a vaccine that saves lives and keeps people from getting very sick. We want a vaccine that can dramatically reduce the number of hospitalizations, intensive care use, and deaths. Kanto Subaro is one influenza expert who said, quote, "The primary benefit of vaccination will be to prevent severe disease." Peter Hotez, a vaccinologist, says Ideally, you want an antiviral vaccine, which COVID vaccines are, of course, to do two things. First, reduce the likelihood you'll get severely ill and go to the hospital. And second, he adds, prevent infection and therefore interrupt disease transmission. And in the US National Public Radio says it quite simply, the primary goal of a vaccine is to keep people from getting very sick and dying. But that is not where the goalposts in the vaccine trials have been set. The FDA announced in June that the standard that it was looking for was either a 50% reduction in lab-confirmed symptomatic COVID or 50% reduction in coronavirus infections. It offered the manufacturers those two options. The companies all chose the first option and set out to test whether their vaccine reduces the risk of symptomatic COVID by at least 30%. For example, in the Pfizer trial, to be counted as a case, you would have to meet this definition. And as you're looking at that definition there, which is taken from the trial protocol, and Serena made mention of these trial protocols, which are public. You can find the links and look at these yourself, because it is very interesting to do that. What I want you to note here is that mild cases of COVID would qualify. The companies were not just counting severe cases, so you don't have to have a serious symptom. So if you're in this trial and you have a cough, and then you get a lab test done to see if you have coronavirus, and if that lab test is positive, you will be considered a case, and the trial will be one case closer to being completed, to reading out its final results. What all this means is that there are certain things that the studies are designed to learn about and certain things that they are not designed to test. So the trials have been designed to test whether they reduce symptomatic COVID of any severity. What they're not designed to provide a definitive evidence, using that phrase that the FDA had earlier, I shared, they're not designed to provide definitive evidence regarding whether they reduce the risk of severe COVID, hospitalization, ICU use, or death. They're also not designed to determine whether they reduce the spread of the disease. And they were not designed to determine their efficacy in important subgroups at highest risk, like the elderly and most frail. And so I have been arguing that the trials, because they are still ongoing, until you can argue they still are, or maybe not, we're on this cusp right now, but they're ongoing. Therefore, our criteria for judging efficacy are not what they should be, and therefore they can and should be made more stringent, because the phase 3 trials are not answering the questions that I think are most important. And one issue that I think, one reason why I think this issue went unnoticed is because most people assumed that severe COVID was what the trials were studying. So the NIH, in fact, in a press release about Moderna's trial, even said this. The trial also aims to study whether the vaccine can prevent severe COVID. The trial also seeks to answer if the vaccine can prevent death. That's actually not what the trials were designed to do. And that's what prompted me to begin writing about this. And I started with an op-ed in the New York Times in September. And I followed that up with a longer article in the BMJ on this topic, which showed really that experts did not know what the trials were studying. So you might be thinking, well, okay, but at the end of the day, why does this matter? If you can stop mild disease, then surely that will prevent severe disease as well, because that's how the disease works. First, you get infected, and then you have some symptoms, and then it gets worse. But while that's true for a single individual, that's not the case when you have many people, as in a trial. In fact, you can have a situation in which there's an overall reduction in mild cases of COVID, but no reduction in hospitalizations, ICU use, or death. If, for example, the vaccine works well in reducing disease amongst healthy young people, but the vaccine doesn't work that well in the elderly, you might reduce cases overall, but you may not be reducing hospitalizations and deaths, as those occur mostly amongst the elderly. And I illustrate this point here in this slide, which is just hypothetical. These are hypothetical data to illustrate a point. Let's imagine a trial with 15,000 people per group. 12,000 of those people were low risk, fairly healthy people. 3,000 were elderly, in which you can expect more high risk, frail, and vulnerable people. When you look at these trial results, you can see here that there's a lot less COVID in the vaccine arm than placebo arm, 60 cases versus 150 cases. So that seems like good news, right? When you look at the types of patients making up those cases, you start to see some considerable differences. Amongst those low risk people, we have 120 in the placebo arm versus 32 in the vaccine arm, which is a 73% reduction. But amongst the elderly, there's just a 7% reduction, 28 versus 30. And with numbers that close, that difference could just be due to chance. So over the whole trial, you get a 60% effective calculation. But when it comes to the important elderly group, it's only 7%. This is similar to what we see for influenza vaccine, which does not work as well in the elderly as healthy adults. So one central worry is, does the vaccine protect the frail? That is, those people who would have a serious outcome if indeed infected. Does it protect them as well as it protects the non-frail? And let me just add some more detail here, illustrative detail for this hypothetical, filling in hospitalizations and death figures. You can see that when it comes to severe outcomes, there might be no differences between the groups. Yet these types of data that I have in this hypothetical would meet emergency use authorization and approval standards from what FDA has laid out because of the overall effect against cases. So now let's shift to what's happened in the last couple of weeks with all the 90% effective headlines. And I'll give you the example of Moderna. The company has made some major claims, not just about a reduction in cases, but actually severe cases, hospitalizations, and death. I'm, however, concerned about what they're calling severe. Is it really severe? How severe? As the press release does not mention anything about hospitalizations, ICU use, ventilator use, and there was just one COVID death in the whole trial. So I wrote the article, and you can see in the slide, last week, and it's my analysis of essentially where we are at this point in time in terms of what do we know and what do we not know. There's a lot of things we still do not know. We don't have any quantitative data to say whether or by how much the vaccines can reduce the risk of infection with the virus. Again, that's not what the trials were out to study. So we don't have any information about that. We don't know whether or by how much they reduce the risk of hospitalization, ICU admission, or death. We don't know how they work in children, adolescents, for the most part, because they were not included in the trials. People who are immunocompromised were also excluded from the trials. We also don't know anything about what the efficacy of the vaccine is at 3 months, at 6 months, at 12 months. If the vaccine works like influenza vaccine, the response of the body will wane over time and vaccine efficacy will drop. I also have some concerns about whether all COVID-19 cases in the trial that occurred were all recorded, and I detail that. It's a bit of a complicated argument, but if you read the article, you'll be able to understand it a bit better. The bottom line there, though, is that in order to figure out whether the recording of all cases occurred as it should have occurred, we would need the data to find out. So I want to leave you with two thoughts. First, there's a saying that the devil's in the detail, and there are reasons to be concerned. And full transparency means having access to all the data. And in this unprecedented time, I think we should be demanding nothing less than public access to all the data. Second, if regulators authorize the vaccines, I think that will mean the end of placebo-controlled trials. So if you remember early in my presentation, these phase 3 trials are our last chance to see how the vaccine works in comparison to a placebo. The end of placebo trials means we won't be able to find out whether the vaccines reduce the risk of hospitalizations, ICU use, and death. Really the key questions. But there is a way, and the reason why I'm talking about this is there is a way to get vaccines to people without compromising the trials. And given the demand right now, the urgency in which people are wanting this access to this vaccine, I think it's really important to know that there are mechanisms that will get vaccine to people without authorization or approval. Access does not require approval. And in the United States, it's called an expanded access program, often also called compassionate use. People generally do not know about this, but this could start today if there was an interest. So I think we should and can and should be talking about this a lot more, and be happy to discuss it in the Q&A. Thanks very much for listening.
Let's start with two questions, and I'll try to read them slowly. The first one, what are some of the areas potentially open for abuse or corruption in the approval and rollout process? Is there a risk of fake vaccination certificates or lobbying for first recipient groups? That's the first one. And the second one, which is also related, aside from the accelerated time of approving all these vaccines, what else has been not normal about this process that reporters should be concerned about?
Well, I guess, Serena, maybe you could take the first one in the sense that what is the, there's this idea that when you investigate health, it's all about corruption, but conflict of interest and commercial interest, how they are intertwined with the normal process is not about corruption. It's much more complicated than that.
Yeah, I mean, I think we are a bit back to square one, that in a way here we have right now a bigger problem. I mean, the biggest issue we have here is that we need to reclaim open access to all the data, because we need people like Peter Doshi to be able to analyze the evidence. We can't just rely on a press release. And also, unfortunately, we also know that regulators, they basically do what they can, but we have to be aware that this is a very toxic environment now, because the regulators and the governments, they are very much under pressure, and that's very dangerous. We don't need these guys to feel under pressure to speed up the process, and that's what's happening now. And in terms of other things that you might want to investigate, one big issue, as you also just mentioned, is the issue of conflicts of interest. In the guide, you find some chapters about this. There is a thing called key opinion leaders. These are scientists that normally work at the same time for the governments and also for the industry. This is a KOL, so-called. They are very important. And I'm sure that right now we have also key opinion leaders working behind the scenes, also on these vaccines. So I don't think right now really the big issue, of course there are countries where we do have a big problem with distribution of drugs or fake drugs, whatever. But I think here the biggest issue we have is really to access to the data. And of course, check out the experts that are advising your government, that are talking to journalists. Check them out, check their background. These, I think, are the most important problems. And also maybe, you know, most contracts, apparently, because they are secrets, right? We can't read them. But I mean, they are like pushing for waiving the liabilities of companies, of pharmaceutical companies. So in a nutshell, the governments, the states, so we, the citizen, are going to be responsible if there will be adverse events in people taking, receiving these vaccines. And this is also something maybe you want to investigate. Over to you, Stéphane.
And yes, maybe for the second part of the question, Peter, maybe you could take that on this idea of what is different with the development of the COVID vaccines. What is different from the normal situation? How did it work? How did the world work before COVID-19?
Yeah, I mean, there's a lot to that question. Lots are different in terms of, I think, the amount of attention on this topic. That's absolutely different. So I just speak to one good thing in terms of that attention is that we saw for the first time the trial protocols released for unapproved products while the trials were ongoing. That does not usually happen. And some of what I presented in this talk today is only possible because of that. And that has to do with pressure, and that has to do with attention. That's unprecedented. On the other side, so, potentially some less encouraging aspects to this unprecedented attention is that if you're a regulator right now, and if you spot problems in this application, I can't imagine being in that person's shoes. It must be extraordinarily difficult to express reservations that would say, actually, I don't think this is ready to be approved. I'd like to see additional data. I don't think that's an easy position to hold right now. So, I think this unprecedented situation in some ways cuts both ways. But in terms of the demands that the public should have for transparency, I think one can't make a more legitimate argument. And the traditional demands for data secrecy, like they're commercial in confidence, and it undermines the company's commercial rights, I think those are undercut by the enormous public investment and the public health consequences of these products. And so I don't think we should allow these vaccines to be rolled out and to be to discuss them without full access to the data. I think that's where journalists can really help, is demanding this basic principle of transparency.
Thank you, Peter.
Thanks for that. I think I'll just go slower, one question by one question, because it's easier to answer it this way. So the next question we have is, after the vaccines are approved and roll out, how are they monitored and for how long? And how can journalists find out if there are new adverse effects or issues? Serena, want to start?
Well, I mean, I can only refer again to the guide, because I mean, the purpose of this publication, so timely, is really to share with our fellows journalists how you can do that. So in the guide, you find like a long chapter about how we do assess and monitor safety when a product also is already on the market. I mean, to cut a very long story short, and maybe Peter can say something about this, but here we have also this problem, because I mean, normally, the logic behind the randomized controlled trial is that you have two groups, right? You have the control group or placebo group. And here we have another problem, because first we have a really short follow-up, because everything has been shortened. And the other problem we have is that we know already, and in Peter's article, you'll find more information about this, is that eventually the placebo group will also get a vaccine, an active product. So there is going to be noise in this data anyway. So this is going to be difficult. I think it's going to be difficult, but I think Peter can say something more specific maybe about this.
Sure. So so before a vaccine is authorized, you have your data is coming from trials, and the problem, as Serena pointed out, is the duration of data and the number of people. And so in terms of the duration, right now, half of the people in the trials have been in them for 2 months after their second dose. So the 2-dose course, they're 2 months out. That's not a long time to find things that will happen after 3 months. You can't find them by definition. They haven't occurred yet. The other problem is the number of people in the trial. 30,000 might seem like a lot, but compared to millions or billions, it's nothing. And so when you're talking about rare adverse events, rare side effects, they won't be detected in trials most likely. And so that's when the question comes to, okay, well, what are the systems in place for detecting and acting on these problems that might be only discoverable after the vaccine is given to many orders of magnitude more people? That is a discussion about what is the quality of the pharmacovigilance system, essentially. And there are structures in place that work on pharmacovigilance. There are people whose full-time job is tasked in this. The problem is there are historical examples that are not that old. For example, from 2009, H1N1 with the swine flu, where the system really, you could argue, failed us in a major way. And I've written about this in the case of Pandemrix vaccine, where there were signals that could have been picked up, and in fact, were picked up early on in the days of this by our pharmacovigilance systems, but not acted on and weren't made public. That signaled some major problems with Pandemrix, but it only was this knowledge that these signals were actually picked up only came out through a legal case 7 years after the fact. And so I have big concerns about whether the system is set up to properly monitor this, and whether we'll have enough transparency that what is detected will be made public. So I am worried about that aspect quite a bit. And let me just, briefly, another reason to be concerned with the trials again is the exclusion of certain populations. So I mentioned immunocompromised people. That's a particular concern, because those people won't be excluded from vaccination programs when they're out in the masses. So that's something to watch out for.
Wow, if we only know about the adverse effects 7 years later, it sounds like health journalists are in for the long haul. Okay, the next question is, there were some questions about what information has been made available about the Russian vaccine. Is the testing and approval process similar to AstraZeneca and Pfizer? Any red flags that journalists should be aware of? I guess Peter can answer this better.
I don't know if I can answer that one that well. I know the least about this, this trial, partly because there's no expectation that it will be submitted to the FDA. What I can tell you is I have written the contact that's listed for this trial on the clinicaltrials.gov website, requesting a copy of the study protocol. No response received. It's a Gmail address. It's hard, I can't make any intelligent comment about that trial. I do find it interesting, however, that the US was contrasting its approach to Russia's approach being sort of get the vaccine authorized first and test later. And we said that our approach in the United States is to ensure that the vaccine reduces, serious COVID disease. And I was thinking, actually, no, our trials are not designed to see if they reduce serious COVID disease. So, so there might be more similarities than one might think.
Maybe just a note, Peter just mentioned clinicaltrials.gov. In the guide, you find all the information about this. Basically, is that since 2005, more and more, like if you want to do a study, you need to register it. So this is like a big database where you find all the studies that have been registered because they started. So it's always good to have a look at that to see if a study is going on, in which hospitals or centers is happening and so on. And as Peter mentioned, apparently for the Russian vaccine, you have a Gmail address to contact the study sponsors. So, but this is something you find also in the guide.
Okay, so I guess this is kind of related to the next question. To what extent can these academic articles about drug efficiency be reliable? What is the most important criteria journalists should take into consideration in assessing the reliability of the academic source? Serena?
Well, it's a really difficult question. I mean, I can only read the guide, because I mean, it's not something you can improvise overnight. It's not that like now I give you a reply and you say, okay, so I can assess if this is a good paper or not. The most important thing you need to know, again, is that the design of a study is super important. There are different levels of evidence. If a study, for instance, is an observational study, we know that it's prone to have many bias, confounding factors, noise that can influence the results. So the standard, for instance, is a randomized controlled trial that should be double-blind if possible. So there is a lot in terms of methodology that you need to get really a bit knowledgeable about. And then you can start assessing independently the quality of the evidence. But it's true that this is a terrible time because there is a lot of publishing, and it's very difficult even for people that are a bit experts sometimes to make sense of what is what. So I encourage you really, to read the guide and try to start from there a bit step by step.
Yeah, I can just add that a journal article, I would much prefer it to a press release, but I started an initiative called Restoring Invisible and Abandoned Trials, or RIOT, precisely because some of the literature is not trustworthy and the literature is incomplete when it comes to the publication of clinical trials. So what I mean by that is that not all trials that are conducted are ever published, and the ones that are, many of them are trustworthy, I have no doubt, but some of them are not so trustworthy. And subtle things or major things in that publication might be wrong and have misleading implications. And so the standard really should not simply be, does it appear in a peer-reviewed medical journal, but does it appear in the public with all the data available underlying that study, so that if anybody wants to check, they can check. If anybody wants to learn more, they can learn more. They can have a discussion about the evidence, the results, and the interpretation of the results with light of all the underlying data. So it's not a question of two experts disagreeing and judging them based on their eminence or where they come from, but rather a discussion that can be really based on the interpretation of data.
Thank you. This other question is a bit more technical, so I think it's for Peter. What is your interpretation of grade 3 severe adverse reactions? A significant percentage of these reactions were admitted in both the Pfizer and Moderna press releases and are largely ignored or downplayed by the media. Is it possible that there were at least some grade 4 life-threatening emergency and grade 5 death adverse events? The press releases don't say either way.
Yeah, so in the Moderna press release, they mentioned that 9% of people had a grade 3 myalgia, muscle pain, 10% had grade 3 fatigue. Those could be the same people, they could be different people. We don't know how those Venn diagrams overlap. The percentages were a little bit lower in the Pfizer trial. 3.8% had grade 3 fatigue, 2% had grade 3 headache. Grade 3 means that the adverse event was severe enough to prevent daily activity. My concern, right, the answer is we would need data to really address all those questions, right? My concern specifically about what this, I would say, very high frequency of adverse events, because remember, if a grade 3 adverse event, something that prevents daily activity, is occurring in 1 in 10 people, then a mild or moderate adverse event is occurring in a lot more people. And I believe that the UK regulator now has some labeling like information up that will give us a few more details that supports that point. The point that I'm concerned about is that when mild or moderate reactions are very common, and far more common than they would be in the placebo arm, it opens up the possibility that reasonable guesses could be made about which group vaccine versus placebo a given participant was in. And if the trials were designed so that the participants and the investigators didn't know which of the two you got, a vaccine or placebo, but if the adverse events profile was very different between the two, intelligent guesses might have been possible, in which case it raises questions about the data collection in the trial. And so in that opinion article that I showed that I wrote a week ago, I go into some details about that and also outline exactly what type of data could be analyzed to address that question, to try and probe whether or not unblinding occurred during the trial.
Okay. The next question, is the full data normally released as part of the FDA application process, or will it only happen if the sponsors do it voluntarily? And do either of you know if nursing home patients were tested in any of the trials?
So the FDA has the rights to ask essentially for whatever it wants. There's a general understanding at FDA from speaking with people there that they get everything. What they mean by everything can differ from reviewer to reviewer. The thing I think is safest to conclude is that FDA, compared to the European Medicines Agency and other regulators around the world, the FDA gets more detailed, granular information than any other regulator. And in particular, the FDA has the resources and capacity to analyze patient-level databases of the data that were collected. And that's not something that all other regulators have the capacity to do. Some of the analyses that I mentioned actually about how you could look at potential for unblinding are only possible if you have that patient-level data. Yeah.
About the nursing home patients?
Yeah, I don't know. Serena, I don't know if you know. They have not, to my knowledge, released any information about the location, the setting from which the participants came. Moderna, from what I've seen, has done the best in providing demographic information, which is on their website. So they tell you, for example, ethnicity, they tell you the age groups and what, so for example, 7,000 of the 30,000 people were 65 plus. So they give you some sort of high level statistics, but I haven't seen anything about nursing home, which is, I think, a very important question, because that's where you would expect the most frail.
Yeah, exactly. The next question, as healthcare journalists, how can we balance being skeptical towards all these press releases on vaccine safety outcomes, but yet avoid contributing to vaccine hesitancy or anti-vaccine movements? Serena?
Well, I think that our duty is to dig deeper. Our duty is to question all the statements that are just fed to us through press release. And here I think about pharmaceutical companies, corporations, but I think also about the governments. This is our duty. And I think, again, we need to demand and reclaim the open access to all the data. And we need to explain to the audiences why this matters, why this is important for them, for their safety and health. And at the end of the day, about vaccines hesitancy, I mean, I think the best friend of it is bad science, bad public health policy, and bad journalism. Because I mean, people have also the right to ask questions. Because I mean, we are talking about, basically, that's why we say that vaccines have to be especially safe, because normally they are given to a population of healthy individuals. So the balancing there has to be especially favorable to the vaccine. So it has to be especially safe, because if I am sick, you give me a drug that has side effects, well, I can take this risk because I'm really sick, I need it. But of course, if you're an healthy individual, the bar should be higher. The problem we have here is that the bar has been set pretty low by the regulators in the first place. And what is going on now is just making it worse. So I think we need to address these questions. It's the only way to not go in the direction of the question that was now asked.
All right, and the next question, what role do reporters have in investigating government policy on rollout priorities, like when they roll out the vaccines that have been approved? Should people who have already tested positive or kids receive it last? Like what could journalists do in that case once it's been rolled out? What can they investigate?
I'm not sure. I think, again, I'm sorry, we're a bit missing the point. I mean, the point here, we have a lot to investigate on how these trials have been done. We need the data. We need to analyze the data. We need to analyze the risk and benefit profile. We need to look at the contracts, if it's possible. Normally they are at one point, 10 years ago, it happened. They were leaked to the press. So we could look at the contracts. We need to look at the liability of the companies. We need to do at the financial part of the story, because the public health money is like a cake. So we are spending at the moment billions on this. We need to see how we end up for our public health systems. So I think there are many, many questions. And I'm sorry to be blunt, but I'm not convinced that is a really important point to investigate who's going to get it first. Because I mean, of course, each country, each government, each public health system will try and do the best they can. And they will, we know it already, they will try first to give these vaccines to vulnerable. So we are talking about, again, the old and frail that have comorbidities, care homes, people. Let's remember that when you go to live into a care home, it's because you are old and sick normally. You can't stay home. So this is a very concerning issue, because as we have seen with Peter's presentation, well, we need to know, was it tested on that kind of population? Because they are going to get it first. So these, I think, are the questions that are really important now.
I'm not sure whether we have time to squeeze in one last question. Do individual countries outside the USA and the Europe monitor vaccine safety once the vaccines have started been used? How can journalists get more information on this?
Sorry, can you repeat the question? I didn't understand.
Do countries outside the USA and Europe with the FDA and the Europe Regulatory Authority, do they monitor vaccine safety once vaccines have been rolled out across the world? And how can journalists get more information on this? Do they always have to go back to the source, or in their own countries as well?
Okay, again, going back to the guide, you have a big chapter about drug safety. To keep it very simple, there is a global system for monitoring adverse events. There is a WHO, so World Health Organization agency that is based in Sweden, where all the reported cases are transmitted to. So you have like national pharmacovigilance agencies, normally are the regulators doing the job. They receive reports from the industry, from patients, doctors, pharmacists, whatever. They assess the quality of these reports, and if they find that they are reliable in a way, they transmit them to the WHO Uppsala, is called Center, UMC. So for a journalist, you find in the guide all the information, what kind of data you can have access to, how you can work with that. It's quite a complex story, but there is a big chapter about it.
Okay, anything to add, Peter? And then I hand it back to Stéphane.
No, thank you. I'm trying to answer the questions in the Q&A as fast as my fingers can.
Thank you, Eunice, for taking care of the audience questions. Unfortunately, we have run out of time now. Before we close, I would like to thank GIJN and our audience today. And a very, very big thank you to our two speakers, Serena and Peter, for taking this time. Before we say goodbye, we hope you can join to the next two GIJN webinars, the last ones in 2020, on open source investigations, featuring the extraordinary online search expert Henk van Hess, on Tuesdays 8th and 15th of December at 9 Eastern Time. Watch our Twitter feed at GIJN and GIJN.org for details on other events. Once again, thank you all and goodbye.